This video, we're going to be reviewing cells and transport and biochemistry. This is on slides 18 through 55 in your review packet. So when we think about life, we can think about all the different scales of life. From the very simplest atoms and molecules are not alive, but they do combine to form this basic unit of life, which we call a cell. And then cells can work together. They can form tissue. And tissues work together to form organs. Organs work together to form organ systems or body systems, which then combine all together to complete a full organism. So it's important to understand that pattern from cell to tissue to organ system to organism, from simplest, which is a cell, to most complex, which is the organism. But there are even smaller parts or larger parts. Smaller than a cell are organelles, and then molecules. Larger than an organism would be things like populations, communities, ecosystems. But we call this organization of life cell, organelle, cell, tissue, organ system. And you can see that here in this diagram. And organelles are structures that are found within a cell that have a certain job to do. Okay. We've looked at slides of cells many times, and we've drawn diagrams of cells. And within cells are these structures that have a certain job to do, and they all work and interact to allow the cell to complete its function. So some examples. Here we have a plant cell. Here we have an animal cell. The plant cells typically have things like the cell wall, okay, and chloroplast if it's part of the plant that makes its own food that goes through photosynthesis. Large central vacuole, nucleus. If we're looking at an animal cell, there is no cell wall, right? There's no chloroplast. There's no large vacuole in the middle. So there's differences between plant and animal cells. If we think about the organelles and their function, the cytoplasm, the gel that sort of fills up the cell, allows things to move around. The cell membrane is the boundary between inside and outside of the cell. The cell membrane has many jobs to do. It has receptor molecules in it to allow cells to communicate with each other. It is selectively permeable, allowing certain things to move in and out of the cell. The nucleus contains the genetic material, contains the chromosomes and DNA, which determine how the cell functions and what the cell does. Ribosomes are the small structures where protein is made, where the instructions from messenger RNA are translated into actual proteins. The mitochondria is the organelle found in cells that is where cellular respiration happens, which means glucose is broken down and the energy in glucose is transformed into energy that's found in ATP. Chloroplasts are organelles where photosynthesis happens. Carbon dioxide and water are taken in, and light energy is required, and it allows the chloroplast to produce glucose and oxygen, glucose being the food that producers are going to rely upon. The vacuoles are storage areas, little bubbles within the cell that store things. So the cell membrane separates the cell from the outside, controls movement of molecules, also allows for communication. We can see the cell membrane has receptor molecules on the outside, molecules that are sort of listening for a signal from some other cell through the use of a hormone. The structure of the cell membrane is what we call a phospholipid bilayer. It has these phosphorus heads and these lipids in the middle, and also embedded within the cell membrane are these proteins that have different functions. Some of them allow materials to move through. Some of them are enzymes. They have many different functions, these proteins that are in the cell membrane. And when we have different concentrations of a molecule on each side of a membrane, what can happen is what we call diffusion. Diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. We can see here these molecules in yellow are diffusing and they are spreading out to a lower concentration. Same thing here. We have a concentrated area of solute here, and it's going to equally distribute over time due to diffusion. And diffusion doesn't require any energy. It just happens. So it doesn't need ATP for molecules to diffuse from one part of the cell to another. They always diffuse from high to low concentration. But there are times when things need to be moved the opposite way, from low to high concentration. And to do that takes energy, takes ATP, because molecules don't want to go to an area where they're already crowded, but they can be forced that way if special proteins and energy is added. That energy comes in the form of ATP. We call that process active transport. So it's active transport if molecules are going from low to high concentration, and it's active transport if it requires ATP. If it doesn't require ATP and the molecules are going from high to low concentration, that's diffusion or passive transport. We already talked about the cell membrane, but again, there are these receptor molecules that are embedded on the cell membrane that allow for cells to communicate with each other. And this is really important, for example, in these neurons, like we looked at in the last presentation. These special chemicals released from one cell bind to the receptors in the next cell, and then that causes some change to happen. Many drugs work by blocking the action of the receptor molecules. So living things require a variety of molecules. And we're going to talk a little bit about what we call biochemistry, about those molecules and what they do. And these molecules are needed for the metabolism of the organism. Metabolism is sort of the overall chemical reactions that take place within an organism. You don't have to worry about this slide very much, but you should know that the main biomolecules that are important in metabolism are proteins, which are made of amino acids, carbohydrates, which are made of simple sugars like glucose, and then fats and lipids, which are made of fatty acids and glycerol. All of these biomolecules contain carbon. Carbon is the main ingredient in life. Okay? And we have two terms that we use for different types of compounds, organic and inorganic. Organic compounds contain carbon and hydrogen. Inorganic compounds are missing one of those two. So for example, water, H2O. Water is inorganic. It has hydrogen, but it's missing carbon. Salt, NaCl. It doesn't have either carbon or hydrogen. It is inorganic. Glucose, C6H12O6, is organic. It has both carbon and hydrogen. And many of these molecules that are important in living things are large molecules. We call them polymers. But they're made of simpler, smaller molecules that we call monomers. Okay? And so in living organisms, sometimes monomers need to be combined to form larger molecules. We call that synthesis, building a larger molecule. Other times, a large molecule needs to be broken down into smaller molecules. We call that digestion. So here we have some of the biomolecules. We have the carbohydrates, we have the proteins, we have the lipids, and we have the nucleic acids. One important thing to take note of is the proteins. The shape of a protein is what is so important about it. The shape of a protein dictates how it functions, what it does. Carbohydrates provide energy. They store energy. Sugars are carbohydrates. Glucose is a carbohydrate. Starch is a carbohydrate made of many glucose molecules bonded together. And so in some cases, glucose is combined, many glucose molecules to form starch through the process of synthesis. Other times, a large molecule of starch is broken down into glucose to provide energy. Fats or lipids are the main compound that makes up the cell membrane. Okay, if you remember from when we just mentioned the cell membrane, it's made of what we call phospholipids. Proteins. Proteins are large polymers. They're made of what we call amino acids. The building blocks of proteins are amino acids. And there's 20 different amino acids that can go into a protein. Which amino acids are put in the protein determine how it folds up. They determine its shape, and as we've said, that's what determines how the protein functions. And these proteins are made in the ribosomes. And then after they're produced, they fold up into their proper shape and they do their function. If we think back to genetics, the instructions for how to build these proteins are stored in the DNA. But they're locked in there. They cannot leave the nucleus. They cannot go out to the ribosome. So in order to actually build the protein, the instructions from DNA are copied to a molecule called messenger RNA, which is smaller. It can get out of the nucleus, can go to the ribosome and attach. And once it does that, the ribosome starts to put together the proper amino acids in the proper order to actually form the protein. And when it's finished, this protein will break away from the ribosome, fold up, and be ready to do its job. It's the shape. Again, when you think protein, you have to think shape. The shape of the protein determines how it functions. Here we have an enzyme. As we know, enzymes fit together with their substrate. It's the shape of this active site that is so important, and this enzyme is a protein. Hemoglobin, the molecule that carries oxygen in our blood cells, it's a protein, and its complex shape allows it to do its job to carry oxygen. Just having one amino acid wrong, different, can change the shape of a protein and allow its function to be impaired. For example, sickle-cell disease, one amino acid, this glutamic acid, is changed to valine, and that causes a person to have sickle-cell disease. It means this protein is misshapen. That leads to their blood cells being misshapen, which leads to a whole host of health problems. And because the shape of the protein is so important, anything that changes its shape is going to impact how it functions. If we change the shape of the protein, it's not going to function in the same way. If we permanently change it by heating it up too hot, it can be permanently disfigured and will no longer work. We call that denaturing. DNA is a nucleic acid. Nucleic acids are another type of the biomolecules, and the main example we talk about in nucleic acids are DNA and RNA. And DNA and RNA, they're large molecules, are polymers. They're made of monomers, the building blocks, which are called nucleotides. And the way that DNA stores information, as we know, DNA has all the information that makes us who we are, all the recipes to make the proteins and our bodies and to regulate how those proteins are made. And it has that because in the DNA molecule is a series of bases. They're called nitrogenous bases. And these bases come in four varieties. There's adenine, thymine, guanine, and cytosine. And the order of these bases, it acts almost as an alphabet, sort of spelling out the recipe for how to build a protein. And so the order of G's, C's, A's, and T's that I have in my DNA will be slightly different from the order of yours, and that's what makes us have different traits, different characteristics. And they're going to be mostly similar because they're both humans, but they are going to vary somewhat, and that variation is what makes us different from each other. This is a single nucleotide. A single nucleotide is a phosphate, a sugar, and then one of the four bases. And a whole bunch of these, millions of these, bonded together is what forms an entire DNA molecule. We know that enzymes are extremely important. Enzymes are molecules which allow a chemical reaction to take place that otherwise wouldn't take place. And so Enzymes are made of proteins, and as we know, proteins, it's their shape that's so important. So the shape of an enzyme, as we see this orange molecule here, has a specific shape because it's a protein, fits together with the shape of the molecule it works on, which we call the substrate. So we see the substrate shape matches together with the shape of the enzyme, and the enzyme causes something to change, cause some chemical reaction to take place. Now, in this case, it caused digestion to happen. It caused this larger molecule to be broken into two smaller molecules. Other enzymes might combine small molecules to larger molecules or do a whole host of other activities in the cell. But an enzyme is specific. This enzyme only will work on this substrate because the shape of the enzyme is specific. Again, when we're thinking proteins, we're thinking about shape of the molecule. That's what's most important. Here's another example here. We have an enzyme binding together with its substrate, sucrose, okay? And it's causing that sucrose molecule to be broken up into two smaller molecules, glucose and fructose. And that process is an enzyme reaction that's digesting sucrose. And once that process is over and the products are released, this enzyme is available to use it to be able to be used again. So again, it can bind with another sucrose and this process can repeat. But it's the shapes that are so important. The shape of the enzyme where it fits is called the active site. Okay. And different factors can influence how enzymes work, how well they work. And the two important ones we think about are temperature and pH. So enzymes work best at a specific temperature or a specific pH. We call it their optimal temperature. For example, this black graph shows the enzyme action at different temperatures. And what we see is this enzyme, its optimum temperature, if we look at the highest point on the graph, is about 37. Well, that's human body temperature. Most human enzymes work best at 37 degrees Celsius. This red line shows a different enzyme. And we see this enzyme works better at a temperature of 78 degrees. So this enzyme is actually found in bacteria, heat-tolerant bacteria. And so if we change the temperature much, so if this human enzyme, if we change the temperature much higher, we see the enzyme rate of reaction drops off very quickly. And that's why if a person has a very high fever, it can be dangerous because as temperature increases, these enzymes no longer work as well. Same shape of the graph is usually present when we're thinking about pH. So here in this black graph, the optimal pH for pepsin we can see is a little less than 2. That's very acidic. Pepsin is an enzyme that's found in the stomach, which is acidic, so that makes sense. Trypsin here is an enzyme that's optimum pH is about 8. That's an enzyme that works in the small intestine. So the pH of the small intestine is around 8, so this makes sense. Changing the pH higher or lower for this enzyme alters how this enzyme functions. Energy. All living things need energy. We know this to just stay alive, to complete daily life process. And the ultimate source of that energy is the sun. And some organisms can utilize that energy from the sun directly and use it to produce food molecules. And we call those organisms producers. We call the process photosynthesis. Photosynthesis takes in carbon dioxide gas and water. And when the sun's light's available, it transforms carbon dioxide and water into glucose, food for the plant. And it also releases oxygen. The process of cellular respiration uses that glucose and oxygen and extracts the energy into a form the cell can actually use called ATP. And the byproducts of that are water and carbon dioxide. So these are both processes, photosynthesis and cellular respiration, that deal with energy transformation. Photosynthesis transforms energy from the sun into energy in the glucose molecule. Cellular respiration transforms energy in a glucose molecule to energy in an ATP molecule. And that's the important molecule that cells need. So we see another description of this, photosynthesis, using light from the sun to make sugar from carbon dioxide and water. Plants go through photosynthesis, algae, some protists, some bacteria, and these organisms have special enzymes that allow them to do this. It takes place in the chloroplast of cells. That's where photosynthesis takes place. We could see it here. Chloroplasts are found in the green parts of plants. Okay, the leaves have many chloroplasts, and that's where most of the photosynthesis takes place. Leaves also have openings called stoma or stomates, which open and close to allow gases to get into the leaf so that photosynthesis can take place, but they also close to prevent water loss, because when they're open, water can evaporate from the leaf. So they help to maintain homeostasis by opening and closing at the proper times. We can see here on the bottom side of a leaf, the opening is the stoma, and then on each side is a guard cell, and those guard cells can close or open depending on conditions. And the purpose, again, is to allow gases into the leaf surface so that they can, photosynthesis can take place. Cellular respiration, that other process we talked about, takes that glucose molecule that has energy in it and transforms it into a useful form of energy, ATP. ATP is what cells run on. It's like, just like your cell phone runs on electricity, cells run on ATP. So this process of cellular respiration is really important for all living things, to take energy and transform it into ATP. It happens in the mitochondria of cells. That's where cellular respiration takes place. All organisms go through this process of respiration, plants and animals. So sometimes students get confused about that. They think that in plants only photosynthesis happens. That's not true. It happens in both. Photosynthesis happens in plants to make glucose, but then also respiration happens in plants so that that glucose can be broken down to turn into ATP. The energy can be transformed into ATP so the plant cell can actually use it.